We've seen artificial intelligence make significant strides in predicting the fixed, three-dimensional structures of proteins. The next frontier in this field, however, deals with a class of proteins that actually defy a single stable shape. These are known as intrinsically disordered proteins. In this explainer, we're going to look at a new AI approach designed specifically to map their vast and dynamic nature.
So, here's our roadmap. We'll start by defining these unique proteins and the challenges they present. Then we'll examine the generative AI solution that researchers have developed, see how it was tested, and finally, discuss the validation of the results and what this could mean for the future.
All right, let's begin with the subject of this study, a category of proteins known as intrinsically disordered proteins, or IDPs for short.
Now, when you think of a protein, you probably picture a structured one, something that folds into a very specific 3D shape to do its job. IDPs are different. They exist as a whole ensemble of different shapes, or conformations. They're constantly shifting, and it's this structural fluidity that is actually essential to how they function.
And these proteins aren't rare. Not at all. It's estimated that somewhere between 30 and 50% of all proteins in the human body are either fully or at least partially disordered. This makes understanding them an essential challenge in modern biology.
Their importance really spans both health and disease. You see, IDPs are responsible for critical cellular tasks, things like signaling and regulation. But when they don't function correctly, they're often implicated in some very serious conditions. For example, the amyloid beta peptide is closely linked to Alzheimer's, and other IDPs are involved in Huntington's disease. And because their function is tied to this shape-shifting nature, mapping their movements is a vital step toward designing targeted therapies.
This brings us to the core scientific problem. How do you map the enormous number of shapes an IDP can take? This is where the traditional methods really start to hit their limits.
The primary tool for studying these proteins has been something called molecular dynamics, or MD simulation. Essentially, this method computes the physical movements of every single atom over time, which generates a sample of the protein's possible conformations.
But here's the fundamental limitation: computational cost. As the researchers themselves note, the amount of time needed to comprehensively map out an IDP's conformational space is often just prohibitive. These simulations are extremely expensive and time-consuming.
So to address this challenge, researchers developed the generative AI model. To be specific, they used a type of neural network called a generative autoencoder, which is designed to mine these conformations much more efficiently.
The whole process breaks down into three main steps. First, an encoder network takes the complex 3D structure of a protein and compresses it into a much simpler representation, a vector within what's called a latent space. Second, the distribution of all these simple vectors is modeled statistically. And finally, a decoder network takes new points from that statistical model and uses them to generate a full ensemble of new, physically plausible protein shapes.
Okay, so with the model built, the next logical step was to verify its accuracy. The researchers needed to test if the AI could not only compress the data, but also generate valid new conformations from that compressed information.
The main performance metric they used was something called reconstruction error. This was measured by the root mean squared deviation, or RMSD, which is basically a way to quantify the difference between the AI reconstructed shape and the original one. A lower RMSD score means the reconstruction is more accurate.
A key finding emerged when they tested 3 different IDPs. For all 3, Q15, amyloid beta 40, and TRIZE, the reconstruction error consistently went down as the model was trained on more simulation data. The results also indicated that, as you might expect, reconstruction becomes more challenging as the protein gets larger.
And that's exactly what they saw with the largest protein, ChinZ, which has 64 residues. They identified a clear challenge. The data from just a single MD simulation was too sparse. It created an irregular distribution in that latent space that the AI's statistical model just couldn't learn from accurately.
The solution, it turned out, was to combine data from multiple separate simulations. When they pooled the training data from 12 different simulations of ChinZ, the distribution in the latent space became much more compact and consistent. This gave the AI a clear single-peaked distribution to model, which in turn significantly improved its performance.
So this refined model was then tested against real-world benchmarks. This final section is all about its validation against experimental data and the overall impact of this work.
Looking at a direct comparison for the cheese protein really shows the model's effectiveness. The AI-generated ensemble was used to predict experimental measurements, and while the traditional MD simulation remains slightly closer to the experimental data, the AI-generated results are highly comparable.
And the key advantage here is that the AI can achieve this level of accuracy with a substantial reduction in the computational resources required.
To put this into perspective, let's go back to the A-beta 40 protein. If you want to sample 100,000 distinct conformations using a traditional MD simulation, you're looking at approximately 80 days of continuous processing time.
Now, this generative autoencoder, once it's been trained, can produce that same number of conformations in just 12 seconds. This represents a significant increase in efficiency, and it could enable the study of these complex systems at a scale that was simply impractical before.
Ultimately, this work demonstrates the potential of AI to efficiently explore the conformational landscapes of these disordered proteins. And it leaves us with a forward-looking question. As these methods continue to mature, how will a deeper, AI-driven understanding of protein disorder reshape the future of medicine and the design of new therapeutics?